Welcome to the ICBAS International Webinar Series. It is my pleasure to introduce the speaker of today, Adrian Krainer. Today, and exceptionally, we'll have an hybrid event because Adrian was in Lisbon and he was very kindly accepted to come to Porto and be on site to meet some of the students. Adrian has contributed to ICBAS as he has lectured to the GABA PhD program some years ago, and actually he was an inspiration for some of these students because some of them led to careers in the RNA field. Adrian Krainer is a biochemist. Is internationally recognized for his work on fundamental science in RNA splicing, but also in the development of mechanism-based therapeutic applications, namely in the development of the antisense oligonucleotide Spinraza, which became the first approved drug for spinal muscular atrophy, a genetic disease that causes motor neuron degeneration. He made outstanding contributions to the field with an impressive publication list and been an inspiration for many students and scientists throughout the world. Adrian was born and grew up in Montevideo, but he then went to the States. He took biochemistry from Columbia University and a PhD in biochemistry from Harvard. He went to Cold Spring Harbor, and he has numerous prizes and awards. He has won many honors, and I'm going to list just a few. Very recently, he received the Wolf Prize in Medicine. He served as the president of the RNA Society, and is a member of the National Academy of Sciences, the National Academy of Medicine, the National Academy of Inventors, and the American Academy of Arts and Sciences, and many more. He was awarded the Breakthrough Prize in Life Sciences for his contributions to the understanding of RNA gene splicing process, and in collaboration with Frank Bennett of Ionis Pharmaceuticals, the development of Spinraza, and he's going to tell you about how to go from the bench to the patient, actually. Adrian is a professor at Cold Spring Harbor Laboratories. He has a very successful research group there, and is a deputy director of research at the Cold Spring Harbor Cancer Center. It is a pleasure to host you here at the ICBAS School of Biomedical Sciences. Thank you for accepting to give this seminar on site and online, and the stage is yours.
Thank you very much. Thank you for the very kind introduction. And yeah, this is a different experience for most of us. This is my first hybrid meeting, and actually it would be the first in-person meeting because yes, since March of 2020 it's all been webinars and so on. I think people are looking forward to a change, so it's my pleasure to be here and to tell you about our work on RNA splicing, and specifically the projects that led to the development of this antisense drug Spinraza. So I have disclosures because I have relationships with some biotech companies, including Ionis and Biogen, with whom we collaborated in the development of this drug. So I'll start by introducing spinal muscular atrophy, or SMA, which is a neurodegenerative disease of motor neurons. It's mainly affecting young patients, so pediatric disease, some adult patients too. And it's inherited as an autosomal recessive condition. And the hallmark of SMA is the progressive degeneration of alpha motor neurons in the spinal cord and the lower brainstem. And it used to be considered the leading genetic cause of infant mortality, even though it's a rare disease, but roughly 1 in 10,000 newborns are affected with SMA. In 1995, so this disease was first described more than 100 years ago, but in 1995, the causative gene was identified by Judith Melke and her colleagues, and they named the gene SMN for survival of motor neuron, reflecting the phenotype. But it turned out that there were two closely related genes, SMN1 and SMN2, and so this numbering system was used. The protein that they code for is called SMN, a survival of motor neuron, and that has been functionally characterized. So first of all, these genes are expressed in all cell types. So we still don't know why motor neurons are the cells that are mostly affected when the SMN protein is not present in normal amounts. And regarding the function, the SMN is part of a complex that promotes the assembly of RNA protein particles, including the snRNPs that function in splicing, but also other RNA protein particles. And but there's another function that has been characterized, which is to promote the transport of messenger RNA along the axons of neurons. And so it's not clear which or whether both or maybe some other function is what's limiting in that leads to the disease state. So patients don't have a proper SMN1 gene due to mutations or deletions. They are dependent completely on the SMN2 gene. And this is a gene that's only found in humans due to a gene duplication. However, compared to SMN1, SMN2 is a weak gene. It produces a small amount of functional protein. And so that's why you have a disease state. But without it, you wouldn't even have a live patient because SMN protein is critical for life. And so this is a question of not making enough protein for the healthy state of motor neurons. Also, another characteristic of SMA is that there are different types. We can classify them based on the degree of severity and the age of onset. And that turns out to reflect mainly the copy number of the SMN2 gene. So if there are more copies of the gene, there's a little bit more SMN protein, and therefore the disease severity is lessened and the age of onset is delayed. So here's the clinical characteristics of the three main types of SMA. So type 1 accounts for about half of the newborn patients with this condition. And it's infantile onset. The symptoms present at younger than 6 months of age, sometimes only quite a bit sooner than 6 months. And these patients have very short life expectancy. So there was a natural history study by Finkel and colleagues. They determined that the median time to either death or the need for permanent ventilation was 10.5 months. So many patients survive less than 2 years of age. SMA type 1 patients are never able to sit independently. They're too weak for that. And 80% of them have 2 copies of the SMN2 gene. The rest have 3 copies. But somehow still have the clinical characteristics of type 1 SMA. So types 2 and 3 are later onset after 6 months of age. Type 2 patients have shortened life expectancy, but nothing like type 1. They are able to sit independently, as you see here. Some, but not all type 2 patients can stand also, but they are unable to walk. And type 3 patients who have more or less normal life expectancy, they can also walk. But in all cases, whatever muscle or motor ability they have declines over time. So ambulatory patients eventually stop being able to walk. And so there's progressive muscle weakness, eventually muscle atrophy. There's also skeletal deformities. And so you can see that type 2 and 3 patients tend to have additional copies of the SMN2 gene. So to understand why SMN1 and SMN2 don't behave in the same way, these are genes that arose by duplication in primates. And in humans, they diverged a little bit. There's up to 11 nucleotide differences between the 2 genes. So they're nearly identical, but not quite. Most of the differences occur in the last 2 introns or in the last exon, which corresponds to the 3' untranslated region. And one of the changes shown here is the only one in a coding exon, in this case, exon 7, which is the last coding exon. So here's the natural stop codon. And the transcription of these genes proceeds and then splicing. And in the case of SMN1, exon 7 is constitutively spliced. So more or less 100% of the RNA is properly spliced and then translated to make normal full-length SMN protein. The transcripts from SMN2 undergo alternative splicing. So the majority of them are spliced by skipping exon 7. And so the next stop codon is present in exon 8. So in effect, this means that the protein that results has a different C terminus. So the last few amino acids come from exon 8. And a small proportion of the RNA is spliced by including exon 7. Importantly, so this change was shown in these publications in 1999 to be responsible for that different splicing pattern. In other words, if you take an SMN1 mini gene and change the C for a T, you start seeing skipping of exon 7. And conversely, if you introduce a C in place of a T at the 6th position of exon 7 and SMN2, now the exon is included properly. So clearly there's a signal for splicing here. But in terms of translation, if the exon is included, these are synonymous codons. And so when the RNA includes exon 7 from SMN2, it makes exactly the same protein as SMN1. But the majority of the RNA doesn't do this. The majority of RNA makes the delta 7 isoform, which at the protein level is highly unstable. So that's essentially the genotype-phenotype correlation. In the absence of SMN1, SMN2 makes a little bit of the correct protein, seems to be enough for most cell types, but not motor neurons. And most of the output of this gene is wasted to make a protein that's rapidly degraded. So when we started working on this disease in the early 2000s, we were investigating the signals for splicing that are affected by the C to T change. We wanted to understand that, but also began to think about how to repair this splicing defect at the RNA level. How can we increase exon 7 inclusion to make more SMN protein, which might have a therapeutic benefit? So that was the thinking at the time. This is just another way of recapping what I've said. These are the mRNAs from SMN2, 10% or so. It's full length, includes exon 7, 90% skips exon 7. You can see the different parts of the protein that are coded by the different exons. And so these two proteins differ at the C terminus, but that's enough to make the delta 7 protein very unstable. So we are going to use antisense technology. So let me introduce antisense oligonucleotides, or ASOs. These are short, synthetic, single-stranded nucleic acids that bind to mRNA or pre-mRNA by base pairing to a complementary region. And in binding, if they bind to the right place, they can modulate protein expression through a number of mechanisms. Either depending on how they are designed, they can promote destruction of the RNA, or they can promote they can block, rather, the binding of proteins to the RNA. So they are synthesized as numerous chemical classes. Most of these comprise a phosphorothyroid backbone. I'll show you some structures, some modifications of the sugar, and sometimes a modification of the bases. And the sequence of an antisense oligonucleotide determines to what RNA it can bind and where along the RNA it will bind. It's something that you basically specify when you synthesize the RNA and you choose a particular sequence. Now, if we design them to bind to a pre-messenger RNA in the nucleus, an ASO can affect the resulting splicing pattern. And so that's the property that we're going to exploit here. I'll also point out that the size and chemical characteristics of ASOs prevent them from crossing the blood-brain barrier. So we're going to have to find a solution to that. So the diagram just shows you a synthetic antisense oligonucleotide hybridizing to a complementary region in a messenger RNA. I don't know where the A went. And okay. So the strategy that we utilize is based on the mechanisms of splicing. So how are exons recognized by the splicing machinery? The splicing machinery recognizes the splice sites and the flanking introns, But when you have alternative splicing, sometimes the exon is included and sometimes it's skipped. And so that's influenced by RNA binding proteins. Activators are proteins that bind to the RNA and attract the splicing machinery. And repressors are the opposite. They bind to the RNA and they inhibit the splicing machinery locally. So in our mechanistic work, we found that the C to T change in exon 7 limits the ability of a particular RNA binding protein to recognize the exon. And that's why splicing is inefficient for that exon. And so our strategy is to target the binding of a repressor to basically compensate for that defect. So if you can see here, if we bind an antisense oligonucleotide to an intron where a repressor normally binds, the oligo occupies the binding site, so the repressor can no longer bind. And so that in effect is the approach we're using. The activator still cannot bind here because it no longer recognizes the sequence, but because the repressor fails to bind as well, the net effect is that we get more exon 7 inclusion compared to when we don't have the oligonucleotide present. So this had not been done before. We're looking for a way to use antisense molecules to promote splicing. And so this seems to be an effective way to do that. And so this work was done in my lab by a very talented postdoc at the time, Yemen Hua, who's now a professor in China. And so we did this work starting in 2004 in collaboration with Ionis Pharmaceuticals. And what's shown here are the modifications of the antisense oligonucleotides that I alluded to before. So if you can think of the RNA or DNA structure, but these three colors indicate the chemical modifications that we're using that make these ASOs distinct from RNA or DNA. In particular, this modification at the 2' position stabilizes the molecule against nucleases. It promotes more efficient binding to the RNA target, and it prevents cleavage of the RNA target by RNase H, which would normally recognize a DNA-RNA duplex, but this is no longer DNA, so RNase H doesn't see it, and so therefore the target is not destroyed. Phosphorothyroid refers to these sulfur groups that are present throughout the backbone. They have been found to promote protein binding. And so unmodified oligonucleotides, if you inject them, for instance, intravenously, they get excreted rapidly through the kidneys. But if they start binding proteins like albumin, then they can stay in the body longer. And also they bind to receptors on the cell surface. They can get internalized, or some proportion of the oligos get internalized. So this is also a very important modification. And then another finding over the years in the field was that introducing these nucleic acids into cells, the cell, it looks like it might be a virus of some sort. So innate immunity tends to be a response to these nucleic acids, and you get inflammatory responses, and so you don't want that. But people found that modifying the cytosines with the methyl group prevents that. And so the oligos we use all have these three modifications throughout. And because of that, they behave more like drugs. They're more stable. They're taken up better by cells. And so long half-lives. And we actually don't need to use lipid nanoparticles or that type of formulation as is used in mRNA vaccines, for example. These are just given in buffer. So how do we find an oligo that works like that? And so I'm just going to summarize how we screen for them. I mean, we screen individual oligonucleotides designed with sequences that would predict where they bind. And so, for instance, oligos were designed against the sequence of the exon, which is 54 nucleotides, and every line here shows an individual oligonucleotide. And if I drew them in green, it means they promoted exon inclusion. The ones in red promoted exon skipping. The ones in blue were more or less neutral. And so we learned a lot from this. There were two classes of oligos that had the desired properties, but we also screened the introns, some regions of the introns on both sides. So if you look at all the data, we're focusing on the green oligonucleotides. So there are 4 different regions of the RNA with quite a few oligos that are effective. So we have to pick between them, and the lines that are a little thicker indicate that they work a little bit better. So I could spend a lot of time on this slide, and I won't, but these are known elements for splicing, enhancers and silencers of splicing. So we can understand the behavior of each oligonucleotide on the basis of the types of sites that they are blocking and the types of proteins whose binding they are blocking. One of these green lines here is Spinraza. So we selected it with as a lead molecule for a number of reasons. And it's shown here. So it's binding to the intron after the beginning of after the 5' splice site. It binds to an element that behaves as a silencer of splicing, and it's 18 nucleotides in length. So it binds from the 10th position to the 27th position. And you know, experimentally, we determined that when the oligo is present, it blocks the binding of these proteins, HNRMPA1 or A2. These are RNA binding repressors splicing. But in the presence of the oligo, they won't bind. And so instead, there's more efficient binding of U1 SNRMP, which is the molecule that initiates splicing by recognizing the beginning of the intron. This is the structure of that molecule, and as a drug, it was named nusinersen as a generic name, and Spinraza is the commercial name. So to do these screens, we use cells like patient cells or other cells that express SMN2 and the splicing assays. But then we wanted to move to in vivo to show that these molecules can work in vivo and do something in motor neurons. And so we used mice, namely mouse models of SMA. Mice have one SMN gene. If you knock it out in a homozygous state, then that's embryonic lethal. But a mouse with one copy of SMN is still okay, healthy. And so transgenic mice were made by various groups. This one has 4 copies of the human SMN2 transgene, and but lacks the SMN gene. But this is also healthy. And if you cross these 2 strains, then you get 50% of mice that have 2 copies of SMN2 and no mouse SMN gene, and that's these mice here. By 7 days of age, you can see that they're smaller than their litter mates, which still have 1 copy of the mouse SMN gene, and they only live another 3 days or so. So this is a very severe phenotype that we consider analogous to type 1 SMA. And so we took these mice and treated them on the first day after birth. So at postnatal day 1, we inject the oligonucleotide. And in order to reach motor neurons, we found that if we deliver the oligo directly to cerebrospinal fluid, then from there it can be taken up by neuronal tissues. And so we're essentially going behind the blood-brain barrier. And so in this experiment, we inject just 20 micrograms once, and then 6 days later, we remove the spinal cord and extract RNA or protein from the spinal cord. And then we can analyze splicing here by RT-PCR. And you can see that the control mice, three controls here, have less than 15% of the top band, which is exon 7 inclusion. But after nusinersen treatment, there's more than 80% exon 7 inclusion. So there's a very drastic switch in splicing, just as we were hoping. And you cannot get this much of a shift in splicing unless all the cells, or nearly all the cells, are taking up the oligo, because all the cells are expressing the transgene. So if you only had 50% efficiency of delivery of the oligo, then you would see this much of a switch from exon skipping to exon inclusion. We use a Western blot to check the protein levels, and you can see that the protein goes up about 4-fold. And in this experiment, that's already enough to give a 70% increase in survival. And then if we give more oligo or repeated dosing or some other method of administration that we also explored, we could increase survival by 25-fold to 250 days mean lifespan. And these mice were not just alive, they were moving around normally. So we also rescue the motor defects. So this was very encouraging, and it motivated full clinical development of Spinraza. And so in this slide, you can see the full timeline of the work. The ASO screens began in 2004, but before that, we were doing mechanistic work to define the target or the mechanism and how we could possibly use ASOs to correct the defect. There was screening, there was lead optimization, mechanism of action experiments to identify the proteins that are normally blocked by this oligonucleotide. But so that work was done in the cell-free extracts and in cultured cells and in mice, and a lot of it in my lab at Cold Spring Harbor. And then Ionis proceeded with tox studies, which have to be done very carefully and are done in rodents, but also in non-human primates, to assess that the molecule is safe in the nervous system and well tolerated, and to come up with a dosing regimen and biodistribution studies, et cetera. And then they started clinical trials in 2011 in November, and then in later stages, they partnered with Biogen to sponsor the phase 2 and phase 3 clinical trials. And this drug was approved at the end of 2016. So just to say a bit more about how the oligos are delivered in the clinic, they're given by lumbar puncture to reach cerebrospinal fluid. What I showed you in mice is called an intracerebroventricular injection, but it's a similar approach, is to reach cerebrospinal fluid, which then circulates throughout the central nervous system. So it's been shown that the initial distribution of the oligo depends on the location and the volume and actually the speed of injection of the bolus that's administered intrathecally or to the lumbar puncture. And so you inject in the lumbar section of the spinal cord, but the cerebrospinal fluid circulates, and so this is called pulsation-assisted convectional transport of the drug. The pulsations of the CSF are induced by the heartbeat and breathing, and they help propagate the drug all the way up through the brain. And from the cerebrospinal fluid, over time, there's transport of the drug into the neural tissue parenchyma, again, throughout the spinal cord and the brain. And then ultimately, the drug is taken up through receptor-mediated endocytosis or micropinocytosis into individual neurons and glia. And so in the clinic, as I said, the drug is given by lumbar puncture, and one good, it's not a trivial procedure, but fortunately, the drug has a very long half-life in the CNS, several months. And so patients really only need 3 times a year to get this procedure. They get 12 milligrams at the moment. There are loading doses that are given more frequently. So 4 injections in the first 2 months to rapidly build up the drug concentration. And then the maintenance doses are 3 times a year. So the clinical trials began at the end of 2011. They enrolled over 300 SMA patients, including infants, children, and adolescents. Those patients who participated in the phase 1 trials but have remained on the drug, some of them have been on this drug for 9 years. That means a lot of lumbar punctures. Infants those trials started 2 years later, so there have been some. They're no longer infants, but they've been on drug for 7 years. And the randomized, placebo-controlled, double-blind phase 3 trials demonstrated statistically significant improvements and clinically meaningful improvements in motor function endpoints and also in survival. And fortunately, and we didn't know whether we could expect this or not for a degenerative disease, motor improvements are seen over time. It's not just stabilizing the disease, but many patients are able to improve over time. The 2 phase 3 trials were ended 1 year ahead of schedule because there was an interim analysis of the data which showed that there was a clear, very significant effect, and so terminated early so that the placebo patients could go on the drug sooner. And I'll mention a trial where patients were genetically diagnosed and then treated before the onset of symptoms, and this gives even better results. So time is very important for a neurodegenerative disease. So here's some data from the clinical trial. This looks at event-free survival. So the time that where there's either death or the need for more than 16 hours a day ventilation. And the bottom is overall survival. And so you can see a very clear separation of treated patients versus the control patients who in blinded fashion received sham injections instead. So it's not perfect. There were still patients who had to go on ventilators, or some patients died, but there's a major improvement compared to the natural history of the disease. So this was done, this is the trial that involved type 1 SMA infants. So I'm going to show you a video of one such patient. If it runs, let's see. Yeah. So this is a patient who was diagnosed very early with SMA. And so he more or less quadriplegic. And so you can see he cannot move his head or his extremities. And he was given 6 months to live. And but the parents didn't give up, and we were able to enroll him in the phase 1 clinical trial for nusinersen. And he started at 6 weeks of age, and so you see over time how he's able to improve. So here he's already moving his head and his arms, and he can grasp an object and pull it towards him. And so these are already things that a type 1 patient wasn't expected to do, like moving his arms like that at 9 months of age. And here he's a year old. I'm not sure if you can hear the sound, but maybe the people online can hear. Then you see him at 16 months, and he can bounce. So he's beginning to do activities that infants or toddlers can do. And he can hold his head up reasonably well. At 20 months, he can stand a bit. And remember that type 1 SMA patients, 2 copies of SMN2, they shouldn't even be able to sit. Now he's 2 years old. So he was using a motorized wheelchair that you can control with a hand, but he no longer needs that. And so a few months later, he was able to do this, which shocked the parents. Okay, so I mean, these videos are from some time ago. So you see here the type of wheelchair that he currently uses, and he can sit on a pony, or he can sit on his dad. And so he's 8 years old now and continues to improve. Okay, so I want to show some results from the clinical trials. This is the phase 1 trial in which Cameron, that kid, participated. There was 17 infants with 2 copies of SMN2, and this is a motor function test where it's called HINE-2. So several motor tasks are scored. Maximum score is 26, and what you can see is that there's a gradual improvement over time. So this is over 1,200 days, And so it's very different from the natural history of SMA. And then the later data are superimposed here from the phase 3 clinical trials. First, you see this curve at the bottom is the 1/3 of patients, 40 of them, who were getting a sham injection for about 12 or 13 months each. And so they're declining over time. That's what the natural history would predict, and it's consistent with it. But the treated patients, 81 of them, the mean changes in motor function, you see pretty much a superimposed curve with the open label trial. Then there was a nurture trial where patients were treated at much earlier in age and based on genetic diagnosis, and that's this curve. So there are 15 patients. So this is an open label study, but the slope is much greater, and this is the type of motor development milestones that you see for normal individuals are very close to that. Very striking difference. Okay, so then there was a separate phase 3 trial for type 2 SMA children. There are different motor function scores, for instance, Hammersmith and ROM, upper limb modules. And you can see again improvements over time, but the sham injected patients either decline or there's no change. So, and these differences are statistically significant, but also to clinicians, the number of point difference represents a meaningful change. This trial involved 126 children ages 2 to 9. So I'm going to show a patient that participated in that trial. That's Emma. Her mother will explain. So this was made in 2016, I think, this video. Yeah, so Emma is now 9 years old and she continues to progress. And a few years ago, she stood in her case for the first time, and you can hear her count because she's still kind of getting used to the idea that she could now stand. She counts to about 100 or so. And so we won't wait for that. She was 2 at the time that she could start receiving the drug, because those are the enrollment criteria for the drug, and it hadn't yet been established that if you do it earlier, it would be better. But you know that was only after these rigorous clinical trials could be carried out. So nusinersen or Spinraza was the first approved drug for SMA. It was also the first drug of any kind to correct defective splicing, and the first time that it was used for exon inclusion therapy. There were oligos that were developed earlier to promote exon skipping for Duchenne muscular dystrophy. This was also the first time that an ASO was administered by lumbar puncture to infants. So there were a lot of obstacles along the way, or things that had to be done for the first time, which is always risky. It was the first approved nucleic acid therapeutic for neurology. And the NURTURE trial demonstrated for the first time that if you treat a neurodegenerative disease, maybe not all of them, but at least some of them, if you treat them pre-symptomatically, you can prevent the onset of the disease, or at least delay it. So far for as long as 5 years, and we hope forever, but time will tell. And this was also the first disease-modifying drug for neurodegeneration. I want to show a type 3 SMA patient. Her name is Sophia. This is after the drug was approved. She lives in Canada. And she, at age 2 and so on, she was ambulatory, so she could walk, but with difficulty. And then she lost the ability to walk. And then by age 4, she started taking Spinraza, and now she can walk. I mean, it's not a perfect gait, but she can ride a bicycle. She was learning how to swim. Her mother sent me an email listing all the things that she could do that she couldn't do before. I hope you can read that. I mean, these are completely different things from what a clinical trial endpoint is, but they're things that we take for granted, and yet for a patient and for the parents, for the family, it represents a meaningful improvement in quality of life. And this is another example of an adult type 2 SMA patient. Phil Harold is a very talented artist. He's a quadriplegic. He could only move his thumbs, and yet he could make digital paintings and sculptures. Very talented. And he also, in Germany, was able to take Spinraza after age 40. And he said that he could tell after a few injections that he didn't feel as cold, that he didn't need, even though he uses ventilation, he could go 6 hours a day without that. And he could move several fingers that he hadn't been able to move for 15 years. Here are some updates, but meanwhile, I'm going to run this video, which to me is the most amazing. So there are many patients like this. So this is Franco, a kid in Argentina, 2 copies of SMN2. He should have type 1 SMA, but he started getting Spinraza after 9 days of age, because he had an older sibling who had the same genotype and had gone through type 1 SMA prior to medication being available. Here at 16 months, he's walking perfectly normally. So this, he wasn't in a clinical trial, but this is very similar to what the NURTURE trial showed. So as of this year, more than 11,000 patients received the drug. It's been approved in over 50 countries. Including Portugal. This type of finding I think you can see that newborn screening is very important, because you need to do this treatment super early to get the most dramatic effects. And so in the US, 38 states are already including SMA and standard newborn screening. And so that encompasses 85% of newborns. Biogen is testing higher doses of nusinersen to see if they can achieve even greater efficacy, and potentially that, I think, could lead to a reduction in the frequency of injections. And there have been since then 2 other approved therapies for SMA, gene therapies, Zolgensma, and a small molecule, Evrysdi. So do I have any time left? Okay. Yeah, all right. So the thing that I'm going to skip this, but just to quickly mention that we're working on other diseases, and to exploit this type of approach, we think ASOs that modify splicing can be applied in different ways for different diseases. We're working, I'll just show a couple of slides on another neurodegenerative disease called familial dysautonomia. But we're also working on liver cancer, cystic fibrosis, and this is a pediatric brain cancer. And these are all preclinical projects that we have pretty promising results, in my opinion. A company I co-founded, Stoke Therapeutics, is working on Dravet Syndrome, which is a genetic epilepsy. And they already have a candidate drug in phase 1 clinical trials. This example, Batten's disease, is a very unique study by Tim Yu and his colleagues, where they developed an ASO to treat a single patient because she had a unique lesion in a gene, and so they try to do this very quickly so they could treat her, and they seem to have some success, but probably it was a little too late, and so that ultimately she didn't survive. But she, during that time, she had a reduced intensity and frequency of seizures, and so this sort of N of 1 approach is personalized medicine is being looked at for quite a few different applications of antisense oligonucleotides. And there are also approved drugs to cause exon skipping and treat Duchenne muscular dystrophy and alleviate the phenotype, so it's more like Becker's muscular dystrophy. So what we're doing for FD is a disease of the autonomic nervous system that ends up affecting every major system of the body, and has very severe pleiotropic phenotypes in cardiovascular, digestive, hypertension, et cetera. And it's a splicing mutation. So the gene is called ELP1, has 37 exons. And the splicing mutation causes skipping of exon 20, abnormal skipping of exon 20. And the patients are homozygous for this mutation at the 5' splice site. So this is a disease that's almost exclusively found in Ashkenazi Jews. And it's very rare. So also in part because of carrier screening, it's more or less eliminated. New patients being born with this homozygous for this mutation. So, but there are still 310 live patients alive. And so we screened oligos with the same strategy, surveying oligos in and around the exon that's getting skipped, finding oligos that promote exon inclusion, and then testing them in mice. Here, first we test them in patient fibroblasts. So this is a normal patient fibroblast. You only get the normal band, but in a fibroblast-rim patient, you can see skipping of exon 20. And then with our lead molecule, we can increase the top band. So we're promoting exon 20 inclusion. Therefore, there's a higher level of the ELP1 protein being made. When the exon is skipped, the mRNA is degraded, and it's a truncated protein results that's not functional. And so we obtained transgenic mice from Sue Slagenhaupt's lab, and they carry the human gene with the mutation in addition to the normal mouse gene. So there's no phenotype, but we can study target engagement. So we can see that in the presence of the oligo, we can, in a dose-dependent manner, increase exon inclusion in brain and spinal cord. And we can also look at the protein using an antibody that recognizes the human protein, but not the mouse protein. And then, so again, we see dose-dependent improvements. So we think this drug is ready to give to patients, but at first one has to show that it's safe. What encourages us is that the chemistry, the chemical modifications are exactly the same as Spinraza's. The sequence, of course, is completely different, and it's 2 nucleotides longer. And our challenge was to convince biotech or pharma to invest in clinical trials for such a rare disease. So for instance, Biogen spent $1 billion in clinical trials and all the associated regulatory affairs and so on to develop Spinraza, and they cannot do that for a very rare disease when so few patients would benefit. However, Ionis started a foundation called NLOREM to facilitate ASO drug development for very rare conditions, typically 1 to 10 patients. And we have discussions with them, and they have agreed to help with the toxicity studies and development, large-scale synthesis, and so on for FD. And so we're hopeful that by next year, a clinician named Horacio Kaufman at NYU will be able to dose the first FD patients with this or a very similar molecule. And so that brings me to the end. So I want to acknowledge the people in my lab who worked on SMA. including the early mechanistic work and then the drug development, especially Yemen Hua, who did the lion's share of that preclinical work. 3 students who worked on the familial dysautonomia project. And both projects were done in collaboration with Ionis, especially Frank Bennett and Frank Rigo. I mentioned Sue Slagenhaupt and Jim Pickle, who provided FD mice, and Dr. Kaufman, who is a clinician treating FD patients, and sources of support, but also patients and their families and the clinical trial teams. And thank you, and I'd be happy to take your questions. Thank you. Thank you.
I don't know, should I go to the chat? Do we have questions? Yeah.
Of course. First of all, congratulations, and it's just so wonderful to see your engagement with the results. I think it must be so gratifying to go from very basic slicing to seeing the kids working. I think that this is really wonderful.
Thank you.
I have lots of questions. Some are basic, some are more applied. But I'll let you one that if nobody else wants to ask questions, you know.
Okay.
One of the things that appeals me is that even in the later injection,
Yeah.
so this is another genetic disease where you lose motor neuron.
Yeah.
But it appears to regenerate.
Right.
So these kids appear to, even if you do the 40-year-old, it looks as if you have some sort of re-engagement of regeneration.
Yeah.
Is that happening?
Right. Yeah. So I'm not a neurologist, so I have a hard time, saying for sure. But in discussions in the field, the thinking is as follows: that the motor neurons that are gone, and so we're not going to be able to regenerate new motor neurons and all the path finding that's normally done in development. But the remaining motor neurons are now healthier. And so one of the things that can happen is that they can multi-innervate more muscle fibers. And I think so that can be demonstrated electrophysiologically, and at least, and some of that work has been done. So you get better function of the remaining motor neurons, and over time actually leads to the muscle beginning to function better. Of course, patients do physical therapy and so on. But there's a limit to what can be achieved. But like I said at the beginning, we didn't know maybe there's a decline, and if you treat, you can stabilize. But in fact, we do see this improvement in many patients. And so it's just somehow that the remaining motor neurons are able to function better. That seems to be what's happening.
Is there any cognitive?
For SMA?
Yes, because this is a model.
Right.
Therefore, you might not expect cognitive dysfunction.
Yeah, there aren't. I mean, in fact, if you meet SMA patients, they're just amazing, and the clinicians, everybody who knows them says that they're above average. The parents, of course, think they're above average, but everybody's parents think that their children are above average. So I think, what they don't know is whether, it may be because being confined to a wheelchair and so on, you have more time to think and read and do things like that. And so they have absolutely no cognitive issues, on the contrary. Actually, I met many families and patients over time, and so they're also very stoic. They don't complain about not being able to do this or that, and so they're just really remarkable people.
Thank you. Great work. It was amazing to see from bench to bedside. I was wondering, in terms of RNA therapeutics.
Yeah.
We have often things in fundamental research to tackle these problems. Why don't we see more drugs like this in the market? What is the bottleneck?
Yeah. Well, it takes time, right? If you look at the timeline, there was 5 years of clinical trials, but to get to that point, you really needed strong preclinical data. So I think every time you have a new platform, there's skepticism, and until there are sufficient technical developments, the early attempts aren't successful. I mean, we were lucky that we entered the antisense field when there was already a very robust chemistry, but that took 10 or 20 years of prior work. And so then having a success like this is an incentive to, so therefore now there's many drugs under development for other neurological conditions in particular, because the properties clearly are beneficial in terms of long half-life and well tolerated and so on. So there's efforts on ALS and Huntington's and Parkinson's, and many other. There have been some setbacks. Don't get me wrong. The Huntington's made it all the way to phase 3, and then there were disappointing results, and so that has to be worked out. And just because it works for one disease doesn't guarantee that it'll work for another, but it's certainly worth pursuing. And there's a lot of activity now in that area. So. But it takes time, so one has to be patient.
I was wondering if the delivery of drugs it's a major problem.
For some tissues, yeah. So for the nervous system, it seems to be very effective. For the liver, it's very effective. The eye, maybe the kidneys, and so muscle, for instance, is more challenging. But there are other chemistries, and there are important improvements that are being attempted, at least conjugates and so on, to get better delivery to tissues that are more difficult to reach. So I think it can be a versatile platform, but it does need constant technical improvements.
We have two on the chat.
Yeah.
Maybe I'll read these two, and then have a couple more in the room. First one, in potential cancer treatments, particularly of disseminated disease, what could be systemic mode of delivery?
Right. Yeah. So the drugs are given, antisense oligos are given systemically. In fact, a lot of what Ionis has done to date involves subcutaneous delivery, and so the liver, a lot of it goes to the liver, but you can reach other tissues, and they have had efforts in oncology. Oncology has been pretty challenging to date. I mean, you have to hit all the cells, or else the tumors come back, and probably combination therapies are the way to go. We're pursuing preclinical models of hepatocellular carcinoma and a form of pediatric glioma, and we have good results in mice. I'm very encouraged by it, but we still have a lot left to do before it gets to clinical trials or something like that. But the Duchenne drugs are given intravenously. Generally, when you do systemic delivery, the half-life is not like what we see in the CNS, so you need it more frequently, but it's also simpler than a lumbar puncture. And of course, the amount of drug you have to give is much higher. But I think there is potential to reach tumor cells and so on through systemic administration.
One more, great talk, thanks. Knowing that SMN proteins function in splicing, do you know if patterns of splicing and alternative splicing are altered through the treatment of SMA patients with spinal disease?
Right. Yeah, it's an interesting question. So SMN, I don't consider it a splicing factor, but it participates in the assembly of SNRs, which are splicing factors. And so there is a bit of a feedback or a loop there. So Michelle Hastings' lab showed years ago that the levels of SMN, so when you go over a threshold to make too little SMN, then you get more exon skipping than before you reduce SMN, and the reciprocal is also the case. That's not, you don't get huge changes in exon inclusion, but you get some modulation of that, and possibly other splicing events change as well. So we have done a little bit of studies of splicing changes in the mouse models, but I think it's been difficult to separate changes that are due to the levels of SMN per se from changes having to do with the mouse going from an unhealthy state to a healthier state. You get loss of splicing changes because of that. And that also makes it hard to look at potential off-target effects of the drug, which I forgot to mention. What I wanted to say is the drug binds to 18 nucleotides in intron 7, and there's no other sequence in the whole genome that is a perfect match. And so we think because of that it's unlikely to cause off-target splicing effects, but there could be some. But on the other hand, the drug doesn't seem to have toxicity at the levels that is used, and so therefore, if there are off-target effects, there's nothing very serious.
The videos were very clear. So.
Right.
Thank you very much for the presentation and for the results. I would like to make two questions. The first one is, does mRNA as drug have some kind of side effects? And the second one is, what are the next steps for splicing?
For splicing, for the field. Yes. Well, so many of you, as I have, probably have received an mRNA vaccine, and so we all wonder, okay, what might this do? I think it's very safe. I mean, I think before it got to the point it wasn't a pandemic, I was wondering myself, would I want to get an mRNA drug or a conventional drug? I mean, after living with the pandemic, I didn't hesitate, and I'm not going to hesitate to get a booster and all that. I think that the issues are, to avoid innate immunity and so on. And so the mRNA drugs have modified nucleotides, in particular pseudouridine or N-methyl pseudouridine, which is similar to the idea of putting 5-methyl group in the cytosine. So it's just to avoid innate immune responses, inflammatory responses. And so those are things that Pfizer, BioNTech, and Moderna worked out, and the academic labs, leading to this. But the pandemic really accelerated these efforts, and so that's one had to wonder, has enough been done to make sure that it's safe? Well, now, you have, I don't know, 1 billion people or something that have been vaccinated. We seem to be doing okay. And so the splicing field, what's next in the splicing field? Well, there's lots we don't understand. So the machinery is very complicated. There's 170 proteins, more or less, and 5 RNAs. That's the splicing machinery. So do we understand what all of them do? Not really. For some of them, we know quite a bit, and for others, it's so people need to continue to study this. There are new structural approaches, cryo-EM, that have been very informative, but they sort of suggest many new things that one can do to better understand the mechanism. We have in our genomes 200,000 or 300,000 introns, and so understanding how the splicing machinery can tackle that with fidelity has always been difficult, but it's very challenging, but very interesting to study.
So the challenge for what would the fun? I have one more and another one there, and then we'll stop. But let me just ask something.
Yeah.
For the students, if they were fascinated by your talk, and if they want to pursue life in science, in molecular biology, RNA, whatever, what advice would you give them?
Yeah, well, you can see that there's opportunities. I've studied RNA splicing my whole scientific career, so including my PhD, and just like you've studied polydenylation for so many years. So I've been at the same institute 35 years, but we go to work every day thinking, so many things we don't know, and we want to better understand. And now we also see that we can apply this knowledge to improve people's health, and there's just so many diseases, and so it's interesting, and I enjoy that our lab is doing both the basic side of things, and we haven't stopped doing that because the new insights that we get from mechanism can inspire new applications, new ways to solve defects related to disease, and so it's fun to do that. I think I remember when I was starting out, always thinking, all the interesting stuff has been done already. We know the structure of DNA, and we know this, but there's always lots in nature that people have no idea, and then it's going to be discovered, like CRISPR-Cas9 systems or RNAi, and then then you get the fun also to see how do I apply that, for therapeutics or to improve crops or whatever. So there's plenty left in nature to be excited about, to discover potentially.
Yeah, so just the joy and curiosity, right?
Absolutely.
One here, great talk from Lilian, much and awesome results. Thank you. What do you think is the key to such a great success of Spinraza? Maybe so many years of fundamental research, but it seems in other cases of AON, such as for Duchenne, the effectiveness is quite different.
Yeah, I mean, that is true. And I think we were lucky there. I mean, I think part of the success, a big part, is that we had an academic industry collaboration with complementary expertise, because, we didn't really know pharmacology, and so we had to learn. And industry splicing wasn't like at the under radar or barely under radar, but so these things come together, and that's good. I think that the Duchenne molecules, it's a bit controversial, how effective they can be. And so part of that can be the nature of the disease. And so maybe we were lucky in that sense, that SMA can be prevented and to some extent reversed. And I don't know that Duchenne is on the same boat in that regard. I think another challenge is that there's a huge amount of muscle, and you have to deliver oligo to that. And they use a different chemistry, but I think it's still limiting how much oligo can get into muscle. And so they have been trying to improve on that. And there's a lot of effort trying to improve the delivery. For instance, conjugating oligos to antibodies that target receptors in muscle, and then get maybe more efficient uptake of the oligos so that they can then change the splicing and so on. But to start out, they're dealing with heterogeneous sets of mutations, and so it's different disease, and they're trying to bypass frame shifts by skipping exons. So that gives a protein with function, but it's not going to be as good as the wild type protein. Whereas in SMA, we're able to make more of the normal protein. So those are the kinds of differences that I think could account for different efficacy.
One last question.
I don't know the question, but I wanted a different comment. So she asked me a question about splicing. That's the next challenge. And you said, there are so many challenges.
Yeah.
One of the things that appears to me, the major challenge.
Right.
What you now was thinking, but could you use the same approach that you use with your antisense oligos to identify proteins that regulate alternative splicing? So that you take a cell line which skips an exon, and then you treat it with the different oligos, and then make it to include the exon, and then find out which is the perfect line. Because at the end, it has to be regulated.
Right.
And so what you doing?
Yeah, that is true. So in a sense, I mean, we kind of have done that. If I didn't spend enough time on the slide where you see that our oligos that cause skipping and oligos that cause inclusion, if we spend time understanding how they do that, we can map elements that that the oligos are blocking, and then we could do some biochemistry to figure out what are the proteins that are doing that. So in this case, the key proteins were SRSF1, whose binding is blocked by the C-to-T change, and HNRMPA1, whose binding is blocked by the drug. And so we already were studying those two proteins in my lab. So for basic studies, we studied it because we do a lot of work on alternative splicing regulation. So I think we kind of have the logic. We understand the logic of alternative splicing, but there's lots of elements, there's lots of factors, and so we have good examples of it. So we understand the basic, the whole logic of it, but not in great detail. I mean, this exon 7 in SMN2 is better understood than anything else because people have mutated it, done there's lots of research on it, but we have 200,000 exons that we don't understand as well. So, not all of them are alternatively spliced, of course, but I think that people have to understand that most genes code for more than one protein, multiple isoforms, and also mRNAs that are not necessarily translated, but they serve a regulatory role in controlling the output of the gene. And so if you do a knockout, you generally are not taking out one isoform, you may be taking out all of them. So you're seeing the net effect. But if you really want to understand the gene, you really should spend time studying the different isoforms. Can you understand what they're all there for? What do they do? And then maybe you can exploit that also for therapeutics. Right? Like our work on cancer is targeting normal alternative splicing of a metabolic gene pyruvate kinase M. So it makes these two different isoforms. The enzymes have different properties, and cancer cells are using that one of those isoforms, PKM2, to support the Warburg effect, the aerobic glycolysis. So that could be a vulnerability, and so we use oligos to switch the alternative splicing to the other isoform, and so that's how we're trying to get at and currently liver cancer. I think there's lots of possibilities to do that because most genes use alternative splicing. In most cases, we have no idea what these different isoforms do, but if once that knowledge exists, there's opportunities for therapeutics.
Okay.
Thank you.
Thank you very much. Thank you.
Thank you all.